In this video, we'll be going over oxidation and reduction. If you've been to any of the SI sessions, this is what's called the table. This table will clearly explain oxidation and reduction from alcohols and the reagents used both in the body and in the laboratory. So, as you can see, I've drawn pictures of primary, secondary, and a tertiary alcohol. So, what can we see here? I'll run through it briefly. This is the left side of the table. As we move right, we will be oxidizing. So if we oxidize our primary alcohol, we get to an aldehyde. If we oxidize our aldehyde, we get to a carboxylic acid. The secondary alcohol, when oxidized, goes to a ketone. The tertiary alcohol cannot be oxidized. So remember, as we go from left to right, we are oxidizing. Conversely, if we go from right to left, we are reducing. Now, if you cannot oxidize, you cannot reduce. So, you cannot oxidize a tertiary alcohol to anything, and you can't reduce something to a tertiary alcohol. Now let's see if we can find any patterns. Looking at this primary alcohol, we see that there are two hydrogens attached to the central carbon. This signifies the two steps of oxidation that can occur. The secondary alcohol has one hydrogen, signifying the one step of oxidation that can occur. There are no hydrogens attached to this central carbon here, which is why no oxidation can occur. Furthermore, what can we see here? When we first oxidize this primary alcohol, what do we lose? We lose two hydrogens. Now, when we oxidize again from aldehydes to carboxylic acids, we gain oxygens. And then obviously the converse is true. If we go from carboxylic acids back to aldehydes, we lose oxygens. Right? So we lose oxygens here, and we gain hydrogens here. Right? For the sake of simplicity, let's keep that off the screen. So what can we say here? If you oxidize, you lose hydrogens, you gain oxygens. If you reduce, you add hydrogens, and you lose oxygens. Now let's go through your reagents. For reducing, you have H2, and you're also given NADH. Now, remember, reduction adds hydrogens, so it makes sense that both of these have hydrogens. Your oxidizing reagents, such as chromate and NAD, have oxygens, or at least no hydrogens. There are three types of reagents that you'll use: reduction reagents, oxidizing reagents, and dehydration. The easiest way to remember it is as what we wrote out. Reducing reagents have a lot of hydrogens. Oxygen reagents either don't have hydrogens or have a lot of oxygens. And lastly, dehydration, the only one you know is H2SO4, which has a lot of hydrogens and oxygens, so it stands out. This is what's called the oxidation reduction table. This will clearly help to explain the steps between the different levels of alcohols and the steps of oxidation and how to memorize them. It's also important to remember that in the body exist only NAD and NADH. In the body, these go only one reaction step at a time. In the lab, we have chromate and H2, which go multiple steps. So for example, In the body, we can only convert a primary alcohol to an aldehyde, whereas in the lab, we can go directly from the primary alcohol through the carboxylic acid. In the next video, we will continue our organic reactions.